Hello, everyone. Welcome to the latest ALA webinar. We're really thrilled to have you here. We've got a good lineup today. Today is new data from historic collections. So in a minute, I'll introduce you to our speakers, and I'm really excited to hear what they've got to say. But I think that today's theme was inspired because I think many of us have had an experience where we've been to a museum of any kind, but particularly a natural history museum, perhaps, and just been awed at the kind of things that you see there and the things you can learn about the natural world. And that's an experience many of us share or have experienced at some point. And yet, when you visit a natural history collection, what might not be clear is the extent to which much of the collection is hidden. I mean, natural history collections are simply too large to fit into museums these days. And so behind the scenes is an enormous trove of biodiversity that has been captured and has profound historical and scientific significance. And here today we're going to hear from three speakers who mine that trove of data to provide insights into nature that would otherwise be impossible. Before we go any further and introduce those speakers, I'd just like to acknowledge that I'm broadcasting today from CSIRO Black Mountain, which is in Canberra. I'd like to acknowledge the traditional owners of the land on which we meet, which are the Ngunnawal and Ngambri people, and pay my respects to their elders past and present. Today we're going to be hearing from three scientists around the country. So we've got Ro Aguilar from Monash University. We've got Emily Roycroft, who's from across the road from me at the ANU. And we've got Ollie Berry calling in from Perth, I believe. Hello everyone, thank you for joining us today. I'm really looking forward to hearing what you've got to say and some really exciting talks. And we're going to start with Ro today. Ro, do you have your slides ready to go?
Yay! Hi. Okay, so I'm gonna share my slides and start with the presentation. So, well,
I can see that perfectly, hearing you perfectly. I'll leave it to you. Thank you.
Cool, awesome. So, well, first of all, hello everyone, and thank you for the ALA for inviting me to participate on this webinar. Museum collections are just an amazing source of science that we can work with, and yeah, today I'm gonna take you inside the X-ray world and show you a little bit of this technique and how the facilities, how it can facilitate to study museum collections. So here we are, a fun group of people, of scientists, actually, working on a linkage grant studying the ecology, morphology, and diversification of Australian lizards. But why lizards? Well, we have more than 850 species of lizards here in Australia. It's a very hot spot for lizards, particularly. So we are trying to ask ourselves, okay, how this happened? So they are so diverse, so different. We are the hot spot for diversity. So we thought about, well, how morphological changes led to this high diversity? So when I'm speaking about morphological changes and variations, all these appear across the whole variation of species and groups. So look at this digit modification from geckos. So depending on the habitat they use, they had literally like claws or pads, digit pads. And it even came down to tail shape and functionality of that tail. So either they can be prehensile, or they can store water, or they can store fat. In this case, Nephrodus levis stores water. And well, hippo on my finger, but that's okay. And this marble gecko, he stores fat in his tail. So this is so different. And then we go all the way down to extreme morphological changes, like limb reductions. What do you think that is? it looks like a snake, it doesn't have legs, it doesn't have feet, nothing. No, it's a gecko, it's a prasha inaudita. So all these morphological changes happen across time because all these creatures has to face the different kind of landscape that Australia offers. It is so diverse. So we thought, okay, in order to study how this happened, we needed to take an ecomorphological, my Spanish coming through, ecomorphological approach to understand what the species ecological role is and how the morphological adaptations to the different habitats evolve. So, fortunately for us, here in Australia, we have, oh my God, such a vast fossil records. It's just amazing the quantity of records, fossil records we have. And these facilitate tracking for evomorphological changes of extinct species to compare to extinct species. Even now with this tiny, look at that scale. They are tiny. Those are osteoderms or varani, the goannas, the Australian goannas. So how we are going to put all these together in order to understand the external and internal morphology through CT scanning. So what we are doing is CT scanning is computed tomography. Computed tomography is a non-invasion technique that uses X-rays to create 3D images of an object. And that one there that you see in the screen is a micro CT scanning. So this is on Tracy's platform at the University of Melbourne, which I work for as well. And I scan there things that are bigger as 30 centimeters long. So everything from a couple of nanometers to 30 centimeters long can fit in there. Otherwise, we have to go and use other CT scanners. So as you can see here, I simplified what the CT scanning structure is, but basically we put an animal on the spot, animal, a specimen, a piece of rock. It could be anything that is in the museum's collections. It could be preserved specimens, it could be dry specimens. So over here, the X-ray source will hit the sample with X-rays, and then the X-ray detector will create projection of what it captured. And it is a 360 degrees projections. So it's a 3D kind of image of what is happening. But how do we get down that down into a computer and into a 3D image? So we log into the computer, and it has a reconstruction algorithm that it will put all the slices that the X-ray took from the sample into one 3D visualization image. It's not going forward. Sorry. There you go. So the convenience of this is that, or the advantage is that a non-invasive technique can let us to study museum collections without causing any harm at all to the specimens. And this is really valuable because I can see the outside, the external features of an animal, but also the inside without having to open them or touch them. So for this project in particular, because we wanted to study all the Australian lizard species, we said, okay, how many species we have at the museum collection? So I went there, I checked it on. If we have them, I will scan them at the University of Melbourne. If they were bigger enough, I will scan them at Monash Imaging Centre. If we didn't have them, I need to loan the specimens from other museums. And we are so lucky that South Australian Museum is part of the linkage grant. So it was a really, really quick interaction there, but also because we have many other side projects happening, everybody was interested in the CT scanning. So almost every museum in Australia cooperated with us. So what is literally, I got there to the collection, I grabbed a sample, and I'm showing you a king cobra. This is not a lizard, but this is part of a side project. So after a CT scan, what I get is these 360 degrees image of what I scan, and then we can have we can search for everything. CT scanning give us some kind of versatile tools, like, for instance, look at this. This snake, I scanned this snake. It has a piece, of a skink tail there inside. So that was her or his last meal. But I can see what the snake eats. And another thing is that we can even sex a specimen, because here in the green part of this blue snake, we can see the hemipenises. So because snakes, you can do that with snakes, not with skinks or lizards, because snakes have a couple of scales in the hemipenis. So that is amazing, because we didn't know this snake was male or female, and now we know it's male. So yeah, another example of how versatile CT scanning is. I can go from this specimen in my hand, that is Rankinia dienmensis, is a little dragon, to, after I scan the specimen, I can 3D print the skull. This is the skull of this guy. So it is so versatile for outreach activities and everything without even touching the specimen. We can do these also cool videos, and and it's quite challenging, because you need to ID what is going on there, who is who, and the good thing is that we can preserve specimens or things like this, that is a skeleton, and I scan it, and after I paint it in different colors, just because it was fun, I can put it together, and this snake was broken. This acanthopsis was broken. So this is the snake that tries to show like a viper. We don't have vipers here in Australia. We only have elatids. So then after I scan it and play with the colors, I can put the whole skeleton together. Another amazing thing is that we can see the external features, but also the internal features. And if you take a look at this one, this is an Anegernia caningami, and there, and I stained this specimen in particular, and so now I can see the external features, but also the internal features with high definition, and I can see the different organs, and that is because I stain the specimens. So sometimes at the museums we have a lot of specimens of one same species. So sometimes they can go and get like treated a little bit funky, like I would stain it. So, and then the solution, we can de-stain them and it's going to take a while. But if it is a common species, sometimes we can do that. And so I can account for the size of the organs inside, and this is fascinating. So, and as we can see, well, again, we can see the internal and external morphologies. We can do fascinating videos and isolating. We can also isolate a specific internal structures for comparison across species. And this frill-neck lizard, as you can see there in orange, there's that is the structure, the skeleton of the tongue. So you will go like, oh, how the tongue doesn't have a skeleton. Yes, it does. And in particular, in this species, is conspicuous because they needed to help to frill out the frill neck to keep it up. So it plays around with some structures in the skull, and the tongue relies on this giant and large skeleton of the tongue to keep the mouth open and do the actual display. But so then how do we actually compare shapes? Size is easy. How do we compare shapes? So after getting the CT scans, we export the volume to a program like MeshLab in this case, and we create a mesh of triangles that will cover the whole surface of the structure. This helped us to statistically quantify and compare the shape. And also, from there, I can 3D print the structure. Another way to do this is through geometric morphometrics. And so actually what we do here, we place landmarks, different kind of landmarks, whatever the points we think is going to be remarkable to show or to differentiate between species. And we delimitate these points of interest and create an anatomical wireframe. And then when that is created here, we can compare that across species, and that will give us a statistical difference from shape of skulls between species. And well, that's all I have to give about the use of CT scanning and museum collections. And stay tuned for an upcoming publication to discover what the calcified endolymph is within Australian skinks. That is the orange part in the last specimen. Thank you.
That's wonderful. Thanks, Karen. That's really incredible talk. Mainly, we're going to save our questions for the end. And if anyone, several people have already started putting their questions into the platform, so feel free to add them, and we'll try and get through as many as we can. Our next speaker is Emily. So Emily, feel free to start getting your slides ready. One quick question, though, Ro, which is, do you how many fossil specimens are there that you're trying to scan here? Presumably there's a lot.
Oh, yeah, no, how many? Oh, there are millions. So we work with Queensland Museum mainly, and I've been up there, like literally searching for fossils, and we have so many, but the thing is that what we are trying to do, like these tiny pieces of bones we find, we are trying to allocate to a group, of a taxa, to a group of animals. So it is really easy when you find a jaw of a skink or versus a jaw of a dragon. So it is really easy to identify because one has pleurodont teeth, The other one is like just a blade of teeth. But what we are trying to do is to standardize. So we go, okay, we have a lot of fossils here. Who are they? So we need to identify, we first need to do that with the extant species. So we go like, okay, here we have a pattern. This is how a gecko should look like. This is how a dragon should look like. So whenever we find a fossil, we go like, oh, yeah. So we have a science opening event every year in Queensland for kids and family. And we let them dig in and they bring little pieces of bones to us. And we use our knowledge to try to identify them in that way. Yeah.
No, it's really interesting. And that idea of a museum collection being a reference library will come up again.
Oh, that's it. That's all we have. Like, it is an amazing resource.
Lovely. Well, in which case, on to you, Emily. Thanks for that.
Great. Can you all see my slides okay and hear me okay?
Perfectly, absolutely.
Fantastic. Okay, hi everyone. My name's Emily Roycroft and I'm a postdoctoral research fellow at the ANU. Before I get started, I also want to acknowledge that today I'm speaking from the lands of the Ngunnawal and Ngambri people, and that the work that I'll talk about today derives from specimens that originate from the traditional lands of First Peoples from across Australia. I acknowledge them as the traditional owners and custodians of these lands. Thanks so much to the team at ALA for the opportunity to talk to you all today about my work using genomics from historical museum specimens to reconstruct the recent extinction of native rodents in Australia. Australia has some of the most unique biodiversity in the world, and especially really unique mammal diversity. As well as the well-known marsupials, we also have many species of native bats and native rodents. But unfortunately, Australia also has the highest rate of recent mammalian extinction in the world. So where 1.5% of the world's mammal species have become extinct in recent times, 11% of Australia's mammal species have become extinct. And this number has just been steadily increasing with time. So there's been at least 34 native mammal extinctions in Australia since European colonisation in 1788. And along with the arrival of Europeans came the introduction of feral predators like cats and foxes, as well as extensive land clearing for agriculture and the beginning of bounty hunting for many native species. Native rodents have been disproportionately affected by this recent extinction. They make up 19% of Australia's mammal diversity, but 47% of recent mammalian extinctions. So there's something about native rodents that makes them particularly susceptible to these threats that have come to Australia since European colonisation. The majority of these species became extinct prior to around 1900, so we really don't know that much about their distribution, their population size, ecology, or their behaviour. And I won't list all of the species because there are lots of them, but on the slide here I'm showing some examples from pictures of specimens along the top, and from drawings. Drawings along the top, pictures of specimens at the bottom. So fortunately, these species were collected alive and lodged as specimens in museums before they became extinct. Like these three pictures I'm showing here from the Melbourne Museum, and we can potentially use these to understand more about the biodiversity that we've lost. So a theme of my research and the theme of our webinar today is generating new data from historical collections, as well as being a really rich resource for studying morphology, as Ro has just so beautifully highlighted with all those videos and pictures. Museum specimens can also be a resource for genome sequencing. I think it's really fascinating to think about these specimens being collected in the 19th century, before anybody knew about DNA, certainly not being collected with genome sequencing in mind. But who would have imagined that hundreds of years later, we could be unlocking the genomes of these species? But new technologies do now allow us to do that, to obtain genomic data from very old specimens. And there are some fundamental unresolved questions that we might be able to answer with this genomic data from extinct species. So one really big unresolved question is whether now extinct Australian species were already in decline prior to the arrival of Europeans in 1788, or whether they went from relatively stable to completely extinct in less than 200 years. Data from the Thylacine and the Tasmanian devil suggest that there may have been longer-term mainland declines in Australia due to the arrival of the dingo, and that was around 3,500 years ago. But we have no information about these patterns for any other species. Another question is, what predicts extinction? Has it been random, or is there something about species, their ecology or their environment that can affect their chance of severely declining and becoming extinct? We have some clues from previous studies about this, but we've never been able to place extinct species in a phylogeny with their living relatives. And this is like a family tree for species that tells us how they're all related. And that's really fundamental to asking these sorts of questions. And how evolutionarily distinct are extinct species from their closest living relatives? This is really crucial to inform our understanding of how much unique evolutionary history we've lost. And we can't do this without genetic data from both extinct and living species. So to answer these questions, we can use a genomic approach. And getting that genomic data involved, in this case, subsamples from over 350 museum specimens. And that was a mix of traditional study skins from extinct species and more recently collected tissues from tissue collections in museums of living species. So from each of these samples, we're able to extract and sequence DNA from thousands of exon loci or millions of bases of genome. I won't go into too much on the methods, but you can definitely ask questions afterwards or check out the published papers that describe these methods. But one of the outcomes of all this sequencing and sampling is the reconstruction of a phylogeny, which, like I mentioned before, is like a family tree, but for species. And this shows us the evolutionary history and relationships of species based on genomic data. So what I'm showing here on the slide is a phylogeny of all of the native Australian species of rodents. So you can see the little coloured boxes along the edge of the phylogeny are showing the IUCN status for each of these species. And those red boxes are the extinct species. So this is genetic data from extinct species that have come from specimens up to 184 years old. This is really cool, and now for the first time we have these species in a phylogeny and we can start to try to get at some of our questions. So our first question was whether these native rodents that we lost first to extinction were already in decline prior to European colonisation, or was the onset of their decline sudden and wholly the result of factors introduced by Europeans? So for most species, we don't have detailed pre-European distributional records, but we can get information about their potential population size by estimating the genetic diversity from specimens that were collected just prior to extinction. And that's a measure called heterozygosity. So using this measure of heterozygosity, we can get a minimum estimate for the amount of genetic variation that was present in each of these extinct species before they were lost. So using whole exome data here, I've estimated the heterozygosity for all of the extinct and living native Australian rodent species. So the grey dots that you can see show species that are in currently large populations, and those are more likely to have higher heterozygosity or more genetic diversity. And the gold dots show species that have come from currently small populations, and those are more likely to have lower heterozygosity or less genetic diversity. But you can see in amongst those red dots, those are the extinct species. And they were from specimens collected very shortly before the extinction of each of these species at the dates that I've put in those boxes. And what these results show is that the level of genetic variation in extinct species was comparable to that of living species in large populations. So this demonstrates that extinct species did not have critically low genetic diversity at the time of their last specimen collection. So their decline doesn't predate the impacts of Europeans in Australia. And I think this really highlights how rapidly these species became extinct. So from this, this tells us that they probably had large populations prior to European colonisation and that they very suddenly disappeared as a result of all of the ways that European colonisation changed the Australian landscape. And this is also backed up by anecdotal reports from early naturalists. And I've put some quotes here on the slide that show some of these extinct species were very common up until the late 19th century. So the white-footed rabbit rat was described in 1846 as the common rat of the country by John Cotton. And the lesser stick-nest rat was described as occurring in great numbers by Krefft in 1866. So our next question was, what ecological or environmental factors predict extinction risk in Australian rodents? So with our phylogeny now of both extinct and living species, which I'm showing again here, we can use an approach called a comparative phylogenetic analysis to test this. And we can ask whether extinction and decline is correlated with factors like body size, biome, the density of human inhabitation, or microhabitat. And what we found was that both body size and biome were significant predictors of extinction risk. So larger bodied species were more susceptible than smaller bodied species, and that extinction and decline has been higher in the arid and mesic biomes when compared to the monsoon biome. So why might this be? Well, there are a couple of threatening processes that are really significant for native rodents, but one of the really big ones is predation by feral cats. So it's possible that larger bodied species are just more susceptible to that predation pressure, or they may be more susceptible to competition with invasive rodents, or more susceptible to habitat modification. And these factors likely vary across habitat type or biome. So this provides us with some really important information for future conservation priorities. And the last question that I wanted to focus on today was this question of how evolutionarily distinct extinct species are from their closest living relatives. So this plot is showing the amount of genetic diversity between pairs of sister species. So in grey squares, that's the pairwise divergence between sister species. And then there's a dotted line which is showing the threshold for when it becomes a within species divergence. And among those points, the red diamonds are extinct species. So the first thing to note is that the majority of those red points fall above that species level threshold, and some of them are quite evolutionarily distinct. So when we do the numbers, we see that more than 10% of the unique evolutionary history in this group has been lost to recent extinction. But what you might also notice is that there's one supposedly species level divergence, which is falling out among the within species data points down here below the dotted line. And this is showing the divergence between an extinct species called Gould's mouse and a living species called the Shark Bay mouse. So our results show that these are actually the same species. So this is the Shark Bay mouse on the left, and on the right are photos of a specimen of Gould's mouse that we sequenced. So Gould's mouse, which was previously thought to be extinct, has actually been hiding in plain sight under another name for more than 150 years, thousands of kilometres away from where the type specimen was collected. So this kind of sounds like good news, and it is in a way, because we get to remove an extinct species from the extinction list in Australia. But what our results also show is that the dramatic collapse of this species in less than 150 years. So if you see this little box here, this is the current natural population for the Shark Bay mouse, which is on an island off the coast of Western Australia. But you can also see how wide-ranging its pre-European distribution had been. And this really highlights that species can be potentially in really large populations with broad distributions, and then rapidly decline to extinction when their environment is fundamentally changed. And it was only because of genomic data that we were able to get from historical specimens of Gould's mouse that we were able to reconstruct this. So to wrap up, these stories that I've taken us through today are just some examples of the ways that genome data from historical specimens can help us to answer important questions, and hopefully they show just how much we can learn by studying specimens of species that we've otherwise lost to extinction. And I just wanted to also say thank you to my co-authors on this project who are pictured here, and to all the funding organisations that support our work. Thanks.
Thanks, Emily. That's fantastic. if a humbling presentation, I definitely think, and some of the statistics you showed there are very humbling about how much the Australian continent has changed and the species that we've lost. I'm going to ask you a question, but I'm going to save it for the end of the seminar. I'm going to plant an idea now, which is the last place I heard some of these ideas were in discussions of a certain concept called de-extinction and the relatedness of thylacines to other taxa. So, think a little bit about that, and we'll frame some questions up at the end.
Okay, sure.
In the meantime, though, I thought we'd hand over to Ollie and just have that seed planted for later on. Ollie, you happy to share your slides?
Sure thing.
Lovely.
See that?
That's coming up just fine. Yep, and hear you just clearly. So, over to you.
Okay, so kaya nunakot waju nungambuja. So I'm saying hi from Perth, which is Wajak Noongar country. And thanks for the great talks, Ro and Emily. Actually, as an undergrad, got to spend some time on Bernier Island, helping with the trapping program as a volunteer of Shark Bay Mount. It's very cute and it's a fantastic place. So look, thanks for the opportunity to speak at this session. And I want to introduce to you a brand new initiative, the National Biodiversity DNA Library. So this is something we're pretty excited about, something we think will transform how we do environmental monitoring and management in Australia. And ultimately, we believe will lead to better decision-making in natural resource management. And it makes it really relevant to a really broad audience. And also just want to highlight that it's fundamentally and deeply enmeshed with the world of taxonomy and collections. And so it's highly relevant to the broader theme of what we're talking about today. So I'm going to start by talking about what a big challenge in environmental management that we think the NBDL is well placed to address. And then also dig down a little bit into how it'll do that. And so where we'll begin is with the challenge of managing natural resources in Australia. So natural resources in Australia, we're highly dependent on them. They're hugely valuable, and however you want to measure that, whether it's economic or social or environmental. And as a developed nation, we've got some really well-established protocols and policies and principles upon which we manage natural resources. But it's still really difficult to do. It's a really non-trivial effort to manage natural resources in Australia. Even gaining some really basic distributional data in Australia for species like those that Emily just presented is really hard, and to do it especially in a timely manner at scale. And that's not a uniquely Australian problem. It's a global problem. But I think the magnitude of the continent, the biodiversity, the relatively low human population size makes it difficult to monitor and measure Australia's environment in a timely manner. And then that makes it expensive. And this is a really consistent theme that emerges when I talk to natural resource managers in Australia. And I imagine some of those people online might have similar experiences or understand it. But it's also something that's been quite more formally fleshed out. So these are a bunch of formal reviews that look into the state of the Australian environment and deal with the challenge of measuring the environment. And they all articulate this challenge, whether it's in biodiversity sector or biosecurity sector. They point to the need for accessible data that's more interoperable, more standardized, more authoritative, with which to make really good decisions. Because the information we're able to make decisions on now is often very sparse and slow in coming. So it's not to say that technologies aren't allowing us to do remarkable things in environmental measurement in Australia. And I think of remote sensing as a really great example. But broadly, the challenge of environmental measurement for the purposes of management in Australia at scale and at speed and with high accuracy is really, really hard to do. What a bunch of these reports also point to is the significant and really growing role of DNA-based or molecular-based measurements of the environment, and that's where I want to go now. And that's to talk about something called eDNA. So eDNA is a fast, universal way an accurate way to detect species and map their distributions. And I expect a bunch of people in this audience will have a bit of an inkling about what eDNA is, but just to get us all on the same page, I just thought I'd reiterate the basics of it. And that's that all organisms, us included, are constantly shedding DNA into the environment that we inhabit. we can now, using some of those technologies like Emily introduced, we can collect that complex mixture of DNA that's in the environment, and we can read the sequences. And because all creatures have a unique DNA sequence, we can tell them apart based on the DNA sequences we find. There's a really topical example of that is COVID in wastewater treatment. That's happening right now. That's a very narrow example of eDNA. So look, this is because it potentially offers a universal and with a lot of potential for automation approach to measuring the environment. It's generating a lot of heat and being picked up a lot by government, industry, and even citizen scientists. And as this little cartoon illustration shows, there are a lot of ways you can use eDNA to measure important aspects of the environment that are relevant to how you manage it. It's the flip side is that it's less reliant on surveys conducted by really individuals with really deep expertise in species, deep taxonomic know-how. So it's that reliance on that really small and knowledgeable group of doing really technically challenging observations of nature, which is really what's limited the scalability of environmental observations. So eDNA, as you probably gather from my enthusiasm, is really quite remarkable and useful, and it doesn't surprise me at all that it's getting a lot of attention. But it's not, look, I think it could do a lot more. It's not nearly as remarkable and useful as it might be. And I'm just going to illustrate that by talking through a little bit about the process of how eDNA is done and how we derive information from it. I'm going to start with an example where we're collecting a water sample. We want to measure some biodiversity. So we collect some water in an environment, and this water sample contains the DNA of a bunch of species that inhabit that area. I've just got some little cartoon illustrations of it. So we can use these amazing sequencing technologies and then obtain those unique DNA sequences, G's and C's and T's. Now, that's not very informative in itself, but to make sense of it, we interrogate a database. And this is a library of DNA sequences where we have known species and attach to them their sequences. So if we go to the environment, we find that sequence. Good news is that we can tell what it is. And it is quite a magical process to somebody, I've done this multiple times, and just that sort of something from nothing, those invisible quantities that reveal such rich information. It's just mind-blowing. The thing is, most of the sequences you find in the environment, you can't say what they are. You may be able to say how many sequences are, but you typically can't, for most of them, can't say what species they belong to. And the reason for that is that those libraries that we're interrogating are really incomplete. Now, I don't really have time to go into the technical details of why that is, but the take-home message is that they're actually really hard to generate. The technology to this point to create those libraries is typically relied on very hands-on and sort of bespoke treatment for each different group of organisms. You have to sort of build a little library. And so you've got all sorts of researchers doing this in different ways, creating non-interoperable DNA libraries based on different bits of the genome which are informative. And so this is not a uniquely Australian problem. In fact, it's a global problem, and it's a really long-standing problem, and it's really holding eDNA applications back. But the good news is we think we have a solution, or we're convinced we have a solution actually. And this is some technology developed by Andreas Swick from the Australian National Insect Collection and his team. We've been working on this for about a bit more over 4 years. And they've developed a really highly refined robotic system and workflow. And I'm not going to go into the technical details, but the take-home message is that this technology can generate from any kind of organism, from a bacteria to a banksia to a bat, using a single universal workflow. It can produce a complete set of DNA reference sequences or barcodes or whatever you want to call them, that anybody ever who would want to use DNA to identify species could use. So for the technically interested amongst you, that's the whole mitochondrial genome, whole chloroplast genomes, whole nuclear ribosomal DNA. And with special relevance to this seminar is it's fantastically good on preserved specimens. In fact, it works better on preserved specimens than fresh specimens. So that's the underpinning technology, which we're kicking off a brand new initiative, the National Biodiversity DNA Library, which I mentioned just before. So this is to build a complete set of reference sequences for all named Australian species. Only a trivial question of 190,000 species. But the technology is there to do some really high throughput, and this is a really manageable problem now with this unique technology. So our vision is really to create this as a highly accessible data, usable, and that's really usable, and has the highest standards of metadata and assurance around those species identities. And that will mean that everybody's on the same page when it comes to making DNA-based identifications. Not only will those identifications be really accurate, but it won't be those sort of dark taxa, those sort of invisible things that we know are out there, but we just don't know what they are. And that will mean people can combine data sets using that single currency that's made possible by this sort of authoritative library. And so we think that'll enable eDNA to become even more widely adopted than it is. Just to paint a little picture about how that all hangs together, directing you over to the left-hand side of this ecosystem, we have the underpinning, the collections of natural history, and this it's about mobilizing those decades, centuries even, of taxonomic know-how that are in those collections, using this unique DNA technology to mobilize those identities and lodge them going up to the top into the National Biodiversity DNA Library where they're available and usable. On the right-hand side of the diagram, you've got the raft of users, end users and stakeholders who are using eDNA increasingly to make measurements of the environment. They take a sample, they generate DNA sequences, they want to know what they are, they can interrogate this authoritative National Biodiversity DNA Library, and with that get identities, map distributions, and apply that in biosecurity or biodiversity or even health, whichever sector they're working. Just to get a little bit less abstract, we just did a little pilot study on this using 380 fish specimens that were collected historically in the, about 5 years ago off the northwest shelf of Western Australia. DNA was available, and these specimens were all lodged in the Australian National Fish Collection. And just pushing it through the workflow, this is just before Christmas, virtually all specimens yielded some really nice DNA sequences. And with minimal intervention, 90% of them are showing us that they're going to yield really complete mitochondrial genomes, which is like an order of magnitude more information than existing technologies typically that are being used to generate reference sequences make available. That's just to bring a bit of reality to it. So like all good initiatives, we have a web page where you can find out a little bit more about what we're trying to do, and soon where you'll be able to access data. We have a brand new team, a director, Jenny Giles, who might be online today, so hi Jenny, and a dedicated laboratory and bioinformatics team that are doing data handling. Andreas and his team have actually already built reference sequences for about 20,000 specimens just in the process of developing this technology. So we're really hitting the ground running with a really informative database from ground up. We are working with government, we're working with NGOs and NCRIS partners like the Atlas of Living Australia and Bioplatforms, so they're already partnering with us. But of course, we're building this to solve problems. We're really serious about that, and we're really eager to understand end users and the broader stakeholder needs of identifications in the field. So we've been doing a lot of consultation, and all I'm just showing here are some illustrations of the different sort of dimensions, the different sectors where we think this is really going to have a lot of value. We spent the last 12 or 18 months doing consultations. And part of that consultation is to meet with groups like this, really to socialise the concept, to make sure people are aware of it and can take maximum advantage of it. So this is just to bring it home, really, to talk about, we think the NBDL is an offering that will really transform environmental management or really have massive national benefit, we hope. We don't want it to be viewed as a CSIRO project. We really want it to be viewed as a consortium that's enabled by some new technology that we've developed. There are many people and organisations that can, and ways that people can support it. And I just thought I'd talk about that in a bit of a call to action. How can you get involved and how can you help make this happen? And the first way is partnering with this initiative. As I mentioned, we've already got government and industry partners contributing resources that can bring the NBDL to life and make it succeed. The second is for users. Firstly, just help us understand how you do eDNA, how you use reference sequences. What would the resource look like? How would it work that would really enable you to do your best work? Provide us with feedback. And when you do use the NBDL, cite it, make sure it's acknowledged and gets the credit that it deserves, because that's what will sustain it. And the final one really is really just about socialising. Spread the word. We, as I keep reiterating, we think this is really going to change the face of environmental monitoring in Australia permanently and give greater certainty to how we measure the environment. I hope greater certainty to decision making. These are things that are really much in demand. You can help us make that happen by just sharing the significance of the NBDL with the people who you think need to know. So thanks very much.
That's wonderful, Ollie. Thanks so much. There's so much information there. And look, we've got 7 or 8 minutes left. We've had some questions from the audience. I wonder if you would mind responding to some of them, if that's all right. So I guess a common theme that we've come out is that all of you are generating enormous amounts of information, whether that's from CT scans or genomic analysis or Ollie, it seems like you guys are really industrializing that. Does that generate challenges for sharing that data and reusing it across research? Anyone can take that, by the way.
Yeah, one of our most difficult tasks was to try to understand and figure out how we store and how we share. Because I have done over the past 5 years almost 40 terabytes of data with species scanning. And that is just the raw data. Then I have additional data whenever you work on the scans, they increase in size. So it is a big challenge. So no institution wants to store so much data. And the thing is that, for instance, I work for Monash University, that we have a cloud there. And I also work for Museums Victoria. We also have a cloud there. But whenever the cloud that we have available in Museums Victoria is limited for obvious reasons, we can't just build it up. And so there's a challenge between juggling around the data to make it forever store somewhere, plus sharing. So my colleagues, I have multiple collaboration projects. So they go like, okay, I need this scan, this scan, this scan, and all the institutions have a different way to share scans. So it's still a challenge because we haven't, across Australia, we haven't come across like a common way to share CT scans. Usually we just do it through sending on mail, like a hard drive with scans, and your colleagues get it. So yeah, it is a challenge that we still didn't solve properly. So yeah, that's a big thing.
Anyone else wish to comment on that?
Oh, I'll just say quickly, yes. I mean, sharing data is, I guess, the core business of the National Biodiversity DNA Library. And it is a challenge, and it's often forgotten that real resources are required to build the infrastructure that'll make that possible. And so we're building that into how we, from the ground up, understanding that. And I think, I guess, just to give a shout out to the Atlas of Living Australia, I mean, I think it's through maybe connecting with existing resources like that, where we don't need to necessarily reinvent all dimensions of what the NBDL will offer, because a lot of that, in terms of contextual information that's associated, say, with a reference sequence we're generating, already exists in the Atlas of Living Australia, and perhaps similarly in other resources. So I guess it's increasingly people are looking to network amongst these infrastructures. Yeah, that's just a little snippet.
A follow-up question there then is that, and Emily, you mentioned this in particular, was where your analysis had led to taxonomic learnings, right? So it's not simply a case of taking a documented taxonomy and learning new things from it, but actually feeding back. And you said you found, for example, that a species has been described twice under different names. And so it's it seems like with the more, as this technology grows, that's going to happen more and more, and there needs to be this feedback loop. Is that a challenge for working in this space, it being the interface between molecular biology and taxonomy?
I think so. I think there's often a bit of a lag time between maybe making that discovery and then it being made public or made official. And so we might all know that there are these undescribed species in our particular groups of interest, or that there are species that have been misclassified, but that actually hasn't made it to a taxonomic publication yet. And so, and sometimes that can go on for years or decades. And it's difficult for conservation because when species aren't described properly, they can't be conserved, they can't be given recovery plans. So I think that any way that we can better integrate those initial results from molecular data or from morphological data and push that through to published taxonomy quicker would probably be beneficial for everyone.
Yeah, I'm curious to see where that goes long term. I was also really interested in what Ollie was saying about the use of this data for monitoring, sort of long term, and again, Emily, your talk talks about the rapid changes that can occur if you have a lot of change in the landscape that can lead to a loss of species very quickly, potentially, certainly on geologic timescales. Ollie, can you talk a little bit about the potential for that technology to provide real-time or near real-time information on species distributions and where that technology is at?
Yeah, so that's a good question. I mean, I think one of the appeals of eDNA is its digital nature. Okay, so you don't have to count bristles or measure scale dimensions. It's a single digital method. And so that lends itself to automation. And so I think there's an appeal there. I don't think other than some very high tech and sort of very localized examples of automated measurement of the environment with eDNA, we haven't quite got there yet. But I think that's, if you just think of, I think it's the mirror in health and biomedical science, that point of care devices, that sort of miniaturization of assays. I think that's where things are going. I think there's a lot of interest. I don't think it's been cracked yet. There's a lot of interest in autonomous vehicles that can go out and measure DNA in the environment. So I think that's a horizon on the horizon that's worth deploying because at the moment, the model really is go to the environment, collect samples, bring them back predominantly. I mean, there's some intermediaries, but until I guess we can do things in situ in an automated way, we really won't be taking full advantage of the information, the rich information that's it, and sort of universal language of DNA.
The last point that I promised earlier on in the talk we'd come back to is that lately there's been a lot of discussion of de-extinction in the news, and clearly that's relying on a basis of historical collections. Does anyone have a view on the relative merits of that versus other approaches to conservation or other spending potentially, or is that too big a question?
I would just generally say that I think efforts would, especially with current technology, be better focused on conserving existing species rather than trying to bring back species that are already lost. I mean, the environment could not sustain a thylacine, and I don't know that bringing them back is going to magically fix that. But of course, there are merits to that kind of research as exploratory. So I don't know if anybody else wants to comment.
I think you've nailed it, Emily.
Agreed. I second Emily. We have a lot of species going down the extinction trace, so we should focus on trying to keep those alive with the environmental that we have at the moment, all the technological challenges, and for bringing up another species into the world. Yes. No, I second Emily.
Lovely. Well, humbling end, but a worthwhile one, I think. Thanks again to all three of you for your talks today. They've been just really fantastic. It's been wonderful to see the sort of work that's happening in our research community and in our natural history collections. We'll run another one of these in 2 or 3 months, so keep tuned if you're interested in hearing more from the Atlas of Living Australia. But for now, thank you to our speakers and have a lovely day.